Now that we've gone through Mendel's model, we're ready to apply his model to the experiments that he did to see how the model actually explains his results in the F1 and F2 generation. This will also introduce to us the Punnett Square. So I'm sure many of you have done Punnett Squares before in science classes, so we'll review how to put together, how to also look at a Punnett Square using this particular slide. All right, so let's start at the very beginning. Here we have Mendel's P generation true breeding variety parents. So we've got the purple flowers. The purple flowers are homozygous for the dominant allele. Now, when we're looking at genetics, we typically use a letter to symbolize a particular gene. In this case, we're looking at the flower color gene. We're going to use the letter P to symbolize this flower color gene. If you have a capital letter P, that indicates that you have a dominant allele for that gene. If you have a lowercase letter, that indicates that you have a recessive allele for that particular gene. So the purple flowers, showing the dominant allele and being true breeders, they have to have two dominant alleles. So their genetic makeup is shown here as two capital letter P's, indicating those two dominant alleles. The white flowers, on the other hand, show the recessive trait, and so being also homozygotes, they need to have two of the recessive alleles. And so here you see that they have two lowercase letter P's, indicating those two recessive alleles. Now, when both of these P generation flowers form their gametes, remember they're going to use meiosis, and during meiosis, you separate homologous chromosomes so that each gamete only gets one set of chromosomes. So all the gametes for the purple flowers are only going to get one copy of every gene. And in this case, the only copy or the only version that the purple flowers have to give are the dominant version of this particular allele. So all gametes will have the capital letter P or the dominant allele. Same thing goes for the white flowers, except in this case, it's going to be the recessive allele. So the white flowers, when they form their gametes, they're going to give each gamete one copy of each gene. The same thing goes for the flower color gene, and the only allele version that they have, the white flowers, is the recessive allele, so all their gametes will get the recessive version of the gene. And so now when you cross these two parental generation flowers, the gamete from the purple flowers will fertilize with the gamete from the white flowers, and all of your resulting F1 offspring are going to be your hybrids, indicating that they have one dominant allele and one recessive allele. They're also heterozygotes, right? So all these purple flowers now have one dominant allele and one recessive. Only the dominant allele, being dominant, shows up in their outward appearance, aka the flowers are all purple. All right, so far, not too bad. Now from here is where it gets a little bit more complicated. So now if the F1 generation hybrids are going to cross with themselves, they first need to make their gametes, right? So in this case, again, remember during meiosis, during gamete formation in plants, you're going to have now the separation of homologs. So one homologous chromosome goes to one set of gametes and the other homologous chromosome goes to the other set of gametes. So the gametes will be 50-50. Half of them will get the dominant allele and half of them will get the recessive allele. And so now we want to know, well, when you cross these F1 generation hybrids to themselves, what are all the possible options for offspring that we could get? This is where we have to form a Punnett square. So Punnett square is essentially a very easy, organized way to use an array to figure out what are all the possible combinations of offspring from two particular parents. So the way that you are going to set up your Punnett square, on one side, you're going to list all the possible gamete options for one of the parents. And on the other edge here, on the other side, you're going to list all the possible gamete options for the other parent. And then you're going to cross. So for example, we've got sperm here on the top. So again, sperm can either be 50-50, dominant to recessive. And then on this side, we've got the egg. Again, same idea because these are also F1 generation females now. 50% of the eggs will have the dominant allele, 50% will have the recessive. and then you cross. So this egg could be fertilized with this sperm to give you a homozygous dominant. You can have this sperm fertilizing this egg. Here you have a heterozygote. You could have this egg right here fertilized by this sperm to have a heterozygote. And then you can have this egg also fertilized by this sperm, giving you the homozygous recessive. And here's exactly now where you see that 3-to-1 ratio. For every one white flower, you're going to get about 3 of the purple flowers. And so Mendel's model fits really, really well with the outcome that he saw in his F1 generation and F2 generation offspring. Okay, now a couple more terms that are introduced here on this next slide. Phenotype and genotype. So when we look at the outward appearance of a particular organism, what we're doing is we're looking at the phenotype. This can be something like eye color or hair color, or it can be something maybe you can't visually see, but it could be something like a predisposition to a disease or an illness. And that is going to be called your phenotype. Genotype, on the other hand, is your actual genetic makeup. So which allelic versions of a particular gene do you have? Are you homozygous dominant? Are you a heterozygote? Or are you homozygous recessive? And so it's interesting to see here the F2 generation P's that Mendel produced, where we see that 3-to-1 ratio. The phenotypic ratio and the genotypic ratio are actually quite different in these organisms. So for the phenotype, we get that 3-to-1 ratio, right? We get 3 purple flowers to 1 white flower. But when we look at the actual genotypic ratio, we end up with 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive, or that white flower. And so there isn't a match in the genotypic versus the phenotypic ratio. And this is interesting to note, right? If you were just to look at Mendel's purple flowers in that F2 generation, just by looking at it, you wouldn't be able to tell which of the purple flowers were heterozygotes and which of the purple flowers were actually homozygous dominant. So that begs the question, well, how did Mendel figure out then at the very beginning which purple flowers to use in his parental generation cross? Because he needed to have the true breeders. He needed to have the homozygous dominant. Just by looking at the purple flowers, he wouldn't be able to pick out which ones are the homozygous dominant and which ones are the heterozygotes. So again, Mendel was smart. And what he did is he performed test crosses to identify which of the purple flowers were true breeders and which were the heterozygotes. To do a test cross, you take your unknown genotype flower. So it has a dominant phenotype, it's purple, but we don't know its genotype. It could be homozygous dominant, or it could be a heterozygote. So you take this unknown genotype flower and you cross it to known genotype recessive phenotype flower. And there are 2 possible outcomes for this particular cross. Depending which of the 2 outcomes you get, you know whether your original purple flower was homozygous dominant or whether it was a heterozygote. So let's take a look at the 2 possible outcomes for this test cross. If this purple flower was homozygous dominant, right, and it were to create its gametes, let's say it was the female flower, the gametes that that purple flower could produce, if it's a homozygous dominant, all of those gametes will have the dominant allele, right? That's all that flower has to give. All the gametes will be the same, carrying the dominant allele. If you cross that to the white flower, as is indicated here with the options for sperm, it's a homozygous recessive flower, meaning that all of the gametes, all the sperm, will have a recessive allele. If you do your Punnett Square then for this particular outcome, you will then see that every single one of these offspring will get 1 dominant allele from the purple flower and 1 recessive allele from the white flower, and they will all end up being purple or heterozygotes. All right, so if all of your offspring from this test cross come out purple, then you know that your original purple flower was a homozygous dominant. The other option is, well, what if that purple flower, the original parent, was a heterozygote? In this case, we look on the right-hand side. And for that purple flower being a heterozygote, 50% of the gametes would get the dominant allele, and 50% of the gametes would get the recessive allele. So here, when you cross that heterozygote now to the homozygous recessive white flower, and you do your Punnett square, you'll notice that 50% of the flowers will come out white and 50% of the flowers will come out purple, because 50% of the gametes from the purple flower have the dominant allele to create the 50% purple flowers, and then 50% of the gametes from the purple flower, original purple parent, get the recessive allele, therefore creating the 50% white flowers. So if your test cross comes out with any white flowers or a 50-50 purple to white ratio, you know that that original purple flower had to have been carrying one of the recessive alleles so that you could actually have that recessive phenotype appearing in the results of your test cross.